Hey, welcome back everyone. In this video, as we continue exploring concepts associated with metabolism and energy, we're going to talk about different types of energy. We'll talk about the difference between the two major types of energy being kinetic energy and potential energy, and then we'll talk about how we can apply these types of energy to living cellular systems, and we'll talk about free energy and the concept of activation energy. So first, let's talk about kinetic energy, which is often called the energy of motion. So kinetic energy describes the amount of energy that a molecule, or really any object, has based on its motion and its movement. Like I said, it doesn't have to be a molecule. It could be something like a bowling ball or a car driving down the street. But in living cellular systems, we're really going to be talking about molecules. So kinetic energy, the amount of kinetic energy that a molecule has, is going to be related to the mass of the molecule and how fast it is moving by the following equation. Kinetic energy is equal to 1/2 times the mass times the velocity squared. Now, when you're talking about the mass of a given molecule, this is not going to change unless that molecule becomes chemically transformed through a chemical reaction, which in this case we're not really talking about. So the only thing that can really be variable in this case is how fast the molecule is moving, the velocity. We saw before in chapter 5, when we talked about membrane transport processes, how fast a molecule moves is going to depend on the temperature of whatever solution that molecule is a part of. So when we talked about the factors that affect how quickly diffusion processes occur, we said that increasing the temperature is going to cause all molecules to start moving faster, so it's reasonable to assume that that would cause a diffusion process to happen faster as well. When we eventually talk about chemical reactions, we're going to find that increasing the temperature does also cause chemical reactions to occur faster as well, because it causes potential reactants that need to collide with each other to move faster, and that increases the likelihood and probability that they will eventually encounter and bump into each other. But there is a limit to this, as we will eventually find out, as increasing the temperature too much will actually cause chemical reactions that happen in biological systems to slow down to a screeching halt. We'll eventually talk about why that is. The other major type of energy that we find in all systems is called potential energy. Just as the name suggests, this represents energy that has the potential to do work even if it is not doing so right now. So more or less, this represents stored energy in some form or fashion that has, again, the potential to be transformed into kinetic energy so that it can be doing work. There are several different types of potential energy, each with their own considerations and their own calculations, which we're not going to get too much into. There's gravitational potential energy, which we'll use an example of on the next slide. This is potential energy due to an object's position with respect to the ground. There is elastic potential energy, which you can see illustrated on the right with a spring. As the spring is in a static position, it has no potential energy, but if you compress the spring or stretch the spring, you inject potential energy into it, because if you let go of the spring in either direction, it will snap back into its original position. In those cases, the potential energy you put into the spring is converted directly into kinetic energy, and that is why the spring moves and snaps back into its original configuration. The major type of potential energy we are going to be talking about in biology is called chemical potential energy. This is the energy that is stored in chemical bonds and is not actively doing work, but can be put to work if we break those chemical bonds and release that energy. And then there's also electrical potential energy, which you very often will see in batteries and electrical circuit systems, which is obviously not the focus of this class. Okay, so one of the main things that we are going to be concerned with is when potential energy is converted to kinetic energy and vice versa. A lot of the metabolic processes that we're going to talk about are going to focus on one type of energy being transformed or converted into another. So we are going to use a non-biological example to show you what exactly we mean by this. Consider this example that we have illustrated here. A wrecking ball is suspended a certain height from the ground. This wrecking ball is not moving and therefore is not actively doing work. since it is not moving, its velocity is 0 and its kinetic energy is 0. But even so, even though this wrecking ball is not doing anything and it's not going anywhere, it has potential energy due to its position due to gravity. The potential energy in this case for gravity can be calculated as mass times height times the force due to gravity, which is the acceleration due to gravity, which is 9.8 meters per second squared, meaning that if we release the wrecking ball, its velocity will increase by 9.8 meters per second every second that it falls. So in this current position, with the wrecking ball just hanging here, 100% of its energy is in potential energy form. Now, if we cut the rope that is holding the wrecking ball up, it is going to start plummeting towards the ground, and it will start accelerating again at 9.8 meters per second squared. So the wrecking ball is going to fall faster and faster and faster as it moves towards the ground. At some point mid-fall, the wrecking ball starts to have velocity, so it currently has some amount of potential energy, which again we can calculate as 1/2 times the mass of the wrecking ball times its current velocity squared. It should say squared there. I forgot to put the squared there. Okay, so however, the wrecking ball still has a certain amount of potential energy because it is still not hit the ground yet. Gravity is still pulling it down. The amount of potential energy the wrecking ball has right now is definitely less than when it was much higher up, but it still has potential energy. What is happening here is that the original potential energy is actively being converted to kinetic energy as the wrecking ball starts to fall faster and faster and faster. In the final scenario, if we consider the wrecking ball the moment before it hits the ground, we can approximate and say that the wrecking ball now no longer has any potential energy because the height above the ground is practically 0 right before impact. But right before impact, that is going to be when the wrecking ball has the greatest velocity, so 100% of its current energy would be in the kinetic energy form. But the thing that you need to remember here is that the current amount of kinetic energy the wrecking ball has, even though 100% of its energy is kinetic in nature, this amount of energy is still less than the amount of potential energy that the wrecking ball started with. Recall that we said that any time an energy transformation process happens, a certain amount of energy is going to be lost as heat. So the reason why we are not able to convert all of that potential energy into kinetic energy is because when the wrecking ball falls through the air, the friction of the wrecking ball moving against the air, we call that air resistance, is going to cause some energy to be lost due to heat. So the air around the wrecking ball heats up a little bit as it falls towards the earth. This is a very important lesson for us to learn because when we talk about this sort of thing and we apply it to biological systems, we're always going to see that energy transformations are inefficient. We're never going to be able to conserve 100% of energy in transforming it from one type to another. We are always going to lose a large amount of energy as heat. Okay, now in biological systems, potential energy that we are going to find is going to be stored as chemical energy. This is the amount of energy that is stored up in chemical bonds, and of course this is a type of potential energy. It is energy that is being stored. It's not actively being put to work, but it does exist. It does have the potential to be put to work. So this brings us back to the concept of the food that we eat, whether we are producers or consumers. When we eat these foods, we use catabolic processes, such as those associated with cellular respiration, to break the high energy bonds found in things like carbohydrates and lipids and proteins, and we extract that energy out and we try to convert it into another type of energy. Cellular respiration is going to involve our cells trying to capture this chemical energy from the food that we eat and then repackage it into another type of chemical energy that is tied up in the bonds of ATP. So in this case, we are capturing chemical energy and then converting it into a different type of chemical energy. Of course, like we said, we're not going to be able to harness the full amount of energy in the food that we eat. Some amount of heat will be lost, but what we are able to capture, we will package up into ATP. And then anabolic processes like those that we've seen before associated with photosynthesis, we of course start with molecules that really don't have very much energy built up in their chemical bonds like what we see with water and carbon dioxide. So if we're going to generate molecules that do have a lot of chemical potential energy, that has to be energy that we capture from the outside, and we actually insert that energy into a newly formed chemical bond, like what photosynthesis does in synthesizing something like glucose. Now, as we move on into Chapter 7, which those videos will be coming soon, we're going to see a lot of molecules like what you see here. These are what we can call high energy molecules. So very often in cells, when you have energy transfer processes occurring in catabolic and anabolic pathways, we use these high energy molecules to store up a lot of that released or required energy on a short-term basis. The idea goes something like this. If your cells break down a glucose molecule, which has a lot of energy built up in its chemical bonds, if we just allow all that energy to be released into the environment, almost all of that energy is going to be wasted as heat. Packaging up that energy immediately into some of these molecules like ATP, NADH, and FADH2 allows the cells to maximize the amount of energy they capture and hold on to that energy on a short-term basis until we can figure out exactly what we want to use that energy for. So ATP, we've seen before, is the high energy currency that all cells are going to use. So when we break down the food that we eat, we store up a lot of the energy in these two high energy bonds that you see right here. The one on the left is the one that contains the greatest amount of energy. In Chapter 7, we'll learn that during the later parts of cellular respiration, which we call the Krebs cycle, a lot of the energy that we capture from glucose is going to be stored up in high energy electrons that are carried by these two molecules here, NADH and FADH2, and they can hold on to those high energy electrons again until we can figure out exactly what we want to use them for. Okay, so let's shift gears a little bit and talk about how we can quantify energy in biological systems. Now think about this. We've introduced the general concept of we eat food, we break down that food, we extract the energy into an ATP molecule, and then we use that ATP energy in order to pay the cost for anabolic non-spontaneous processes. Well, that's all fine and well, but how do we determine how many ATP molecules are necessary to power a certain anabolic process or a certain non-spontaneous process? When you think about paying bills and earning money from your job, you don't think about it in terms of, oh, I earn money and then I pay my bills. You think about it in a quantitative fashion. You ask important questions like, how much money am I earning per week? Is that going to be enough to buy groceries? Is it enough to pay my electric bill and my gas bill? You're thinking about things in a quantitative manner. How much money does it take to do this? Well, the same applies to cells. The energy released from one ATP molecule may be enough to power one type of non-spontaneous process, but maybe not others. So in those cases, it would require multiple ATP molecules to provide the energy for something that is more energetically costly. So our ability to quantify chemical energy and potential energy in biological systems is going to be essential for us determining whether the energy is enough to power a spontaneous process or ensure that it is able to move forward. So like I said, this type of quantification would really allow us to determine whether the energy that gets released from one particular molecule of ATP would be sufficient to do something like build one protein. And if not, we would have to start asking questions like, well, how many molecules of ATP is it going to take for us to do that? So the term we use to talk about the change in total energy in a biological system is called Gibbs free energy. This is represented as delta G. The delta in this case refers to the change that is occurring between two time points. The reason we talk about the change in Gibbs free energy rather than the total amount of energy is that by experimental methods, it is nearly impossible to actually get an idea of how much total energy is in the system. But instead, we can actually determine how much that energy has changed. Now, if you're a cashier and a person walks into your store and buys a certain number of items and you check them out, you can determine how much they paid for those items. You're not going to be able to determine how much total money they have to them in their bank account, but you can determine by how much that total amount of money has changed by the transaction that they just did. If they pay for $70 worth of goods, you can say that their wealth has decreased by $70. You don't know how much money they have, but you do know that they're now $70 poorer than they were before. That's the sort of thing that we're talking about here. Okay, so let's talk about how we can go about calculating the change in free energy. We can do it according to this equation here. Delta G equals delta H minus T times delta S. Now, let's define some of these terms because a lot of this is going to probably seem foreign to you. Delta H is called enthalpy. Enthalpy refers to the sum total energy of the system. You're just talking about total energy being total kinetic energy, potential energy, while also considering any changes in energy due to pressure and volume effects, whether it be compression or contraction or things like that. T, of course, represents the temperature of the system. We have to consider the temperature because we've already seen that changing the temperature changes the amount of kinetic energy molecules in the system has. The interesting thing about temperature here is that we absolutely have to express temperature here as degrees Kelvin. So to find degrees Kelvin, you simply add 273 to the number of degrees Celsius. Now, of course, I have made another typo here. 0 degrees Kelvin would be what we would consider absolute zero. Absolute zero would be equivalent to negative 273 degrees Celsius. So I made another boo-boo there, so make sure that you take note of that before you go any further. So negative 273 Celsius would be the same as 0 degrees Kelvin, and that is absolutely the lowest temperature can go. Molecules will stop moving at that point. Human body temperature, which is 37 degrees Celsius, is equivalent to 310 degrees Kelvin. Finally, delta S refers to the change of entropy of the system. We're going to talk about the concept of entropy in a later video when we discuss thermodynamics, but for now you can think of it as the relative amount of disorder or chaos in the system. Okay, so let's talk about using the change in free energy to determine whether a reaction is spontaneous and will release energy, or whether a reaction is non-spontaneous and will require energy. So what you need to look at are these two red horizontal dotted lines. This first line here represents the initial energy of the reactants. This bottom red line here represents the final energy of the final products. When we make a comparison here, we can see that the initial energy of the reactants is much greater than the energy that the final products have. You can kind of think of the progression of the chemical reaction here as going down a steep hill. Because the reactants have more energy than the products, if we allow this reaction to proceed, and for now you can kind of ignore this big bump right here, if we go from here down to here, the delta G is less than 0, which means the energy is being released. The total energy in the system is going down, and that difference in energy is being released. This is how we can define a spontaneous reaction, one in which the change in free energy is less than 0 and is a negative number. We're going downhill. The reactants have more energy than the products. Good example we've seen of that before is the combustion of glucose. We said for sure that glucose has more energy than either of the products, water and carbon dioxide. So that's what makes that a spontaneous process. So in those cases, we call those types of reactions exergonic reactions. These release free energy from the system, and very often that release of energy can be put to work, whether it be packaging into ATP or doing something more direct. Now, if we look at the reaction on the right, we can see that the initial energy of the reactants is actually much less than the final energy of the products. You can kind of think of this as an uphill process. In these cases, the change in free energy is actually a positive number, meaning that energy is going to have to be put into the system. The system is going to have to gain free energy in order to pay the cost for getting this chemical reaction to move forward. So this is how we determine a reaction is not spontaneous, if it has a delta G that is greater than 0. This means that the reaction will not move ahead unless we inject the determined amount of free energy into the system, otherwise the reaction is not going anywhere. In these particular cases, we call them endergonic reactions. Exergonic reactions, exer like exit, energy is exiting the system. Endergonic reactions, energy is entering the system, enter. You can kind of think of it that way. Now, one thing I want to make clear, and we're going to talk about this very soon in a new video when we talk about enzymes, you have to be careful. The word spontaneous does not mean the reaction goes fast. In fact, sometimes it is exactly the opposite. A spontaneous reaction, when we use the term spontaneous, all that means is that the reaction, if we give it enough time, it's going to move forward, okay? So if we say slap down a bunch of glucose on a table and that glucose is surrounded by the oxygen in the air, just because the combustion of glucose with oxygen is spontaneous, that does not mean it's going to happen right away. In fact, it takes many, 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 many years for glucose to spontaneously combust into carbon dioxide and water. So we'll talk about exactly why that is, but just be aware for now, a spontaneous reaction is not necessarily a fast reaction. Spontaneous reactions very often are actually extremely slow. It just means that if we give it enough time, we may be waiting a while, but if we give it enough time, eventually the reaction will happen all by itself. But very often we will be waiting a very long time. Okay, so let's reconsider the equation that we looked at before with the idea that we want to maximize the chances that a reaction will be spontaneous. If a reaction is going to be spontaneous, we saw that the delta G needs to be a negative number. Well, if we look at the other side of the equal sign here, what do delta H and delta S need to be in order to give us the best chance at this reaction being spontaneous? Well, of course, we need delta H to be a negative number. We would prefer that the total energy in the system also decreases. It doesn't necessarily have to be that way, but we would prefer it to be that way to maximize the chance that the reaction is spontaneous. And then the next is the product of T delta S. Now, in this case, we want delta S to be a positive number. The reason why we want it to be positive is because here, delta G is equal to delta H, which we know we want that to be negative, minus T delta S. If you remember some of your elementary and middle school mathematics, if you subtract a negative number, that's actually like you're adding a positive number. So delta H minus T delta S, if delta S is a negative number, we are actually causing delta G to become more positive. So we actually want delta S to be a positive number. And since we define entropy as the amount of disorder in the system, in order for a reaction to maximize its chances to be spontaneous, we actually want the amount of disorder in the system to increase. We want things to become more and more and more chaotic. Okay, so the idea here is that if every metabolic reaction were allowed to reach equilibrium, at which point there is no longer a change in delta G, then cells would no longer have a sufficient amount of free energy to maintain activities associated with life. So what you're going to find out here is that every metabolic process inside of your body is always trying to reach equilibrium. Every system in your body, every cell, every portion of the cell is always trying to get to the point where the delta G for a reaction equals 0. Again, if we get to that point, energy transfer processes will no longer occur, and we will stop living because we will no longer have enough free energy to power anabolic and endergonic processes like protein synthesis and things like that. So to help us with this, We resist that by always having a constant input of energy into cellular systems, either because we consume and digest food or other things. So every type of metabolic process is always in a state of flux so that these chemical reactions cannot reach a delta G of 0. So we ensure that there's always going to be a sufficient amount of energy to power all of our cellular processes. Okay, the last thing that we want to talk about before we wrap up this video is I want to discuss the elephant in the room. Look at this exergonic process that we talked about before. We determined that because the delta G is negative, it's less than 0, this is a spontaneous reaction. But do you remember how I said a spontaneous reaction is not necessarily fast? Well, let's look at why that is. You can probably put 2 and 2 together and figure out that the reason why this is not a fast reaction is because of this big bump that you see right here. That big bump, we're going to learn, is called activation energy. This is essentially the amount of energy that has to be input into the system in order to get the reaction started. So if you think about riding your bike down a hill, if you're just riding straight downhill with no obstructions, yes, you are going to be riding very fast. But if you have to ride your bike uphill before you eventually go downhill, that's going to greatly slow down your progress. So that is what we are considering here. The presence of an activation energy barrier is the reason why a lot of spontaneous reactions are actually very slow. So if we go back and reconsider the potential combustion of glucose with the oxygen that's in the air, this is the reason why if you slap down some sugar on the table, it's not just immediately going to combust into carbon dioxide and water. That reaction has a very large activation energy barrier. So if we want that reaction to actually occur in a timely fashion within seconds or so, we need to introduce something called a catalyst. A catalyst is a type of participant in the reaction. It does not actively participate in the reaction in the sense that it is chemically modified itself, but it does drastically speed up the reaction. So the idea here is that even though the glucose, which has more energy than either of the products being carbon dioxide and water, that activation energy is what has to be overcome. And we are going to learn that what a catalyst does is it greatly reduces the amount of activation energy that is required in order to get the reaction going. So again, if you consider the bike example, if you greatly reduce the size of the hill you have to bike up before you can bike down, the whole process is going to become a lot faster in that regard, and it makes the reaction a lot more likely. So what the activation energy actually means in scientific terms is this is the amount of energy that is needed to get the reactant molecules into just the right three-dimensional orientation, talking about things like their bond angles and their access to each other. You have to get things just right before the reaction becomes more likely. If two molecules that are supposed to react with each other collide, but they don't collide in just the right way, then the reaction will not occur. So a catalyst will ensure that the collision happens in a productive manner. So this is what enzymes are going to do. Your biological catalysts are enzymes, and we have learned before they are almost always proteins, and we will talk more about enzymes in a later video. So the larger the activation energy requirement is in an uncatalyzed reaction, the slower that reaction will be, because again, that represents how much energy you have to put in before the reaction can move ahead. So the idea here is that enzymes will speed up reactions by, again, greatly reducing how much activation energy is required. So accumulating a much smaller amount of activation energy becomes a lot more feasible, meaning we're going to be able to get this reaction going a lot more quickly because we have greatly reduced that activation energy barrier. Getting over the green hill is a lot easier of a task than getting over the purple hill in that regard. Now, this might seem like a very inconvenient thing, but think about this. Without activation energy requirements, any molecules inside of our cells, like proteins and carbohydrates, lipids, ATP, and things like that, these high energy molecules that store their energy up in chemical bonds, Without these activation energy requirements, all these molecules would just spontaneously break apart very quickly and very unexpectedly. So, since the cell is constructed out of materials that, by their nature, should break down spontaneously, your cells would just disintegrate right away and nothing would have any stability. But because of these activation energy requirements, these molecules all maintain some stability and ensure that the only way they are going to break down is if an enzyme is activated and actually causes that breakdown to occur. And we're going to eventually learn that cells have a remarkable amount of control over which enzymes are working and when and how. All right, that's going to wrap up this video talking about different types of energy. So thanks again for your attention. Join us next time and we will have a discussion on the laws of thermodynamics so we can know what kind of rules that energy transfer processes have to follow under every circumstance. Thanks again. See you next time.